Hello everyone, happy afternoon, a very good afternoon. I hope and I believe all of you are doing well. A quick nod everyone, am I audible and visible? Okay, that's great. Thank you so much, Jojoba Binesh, for confirming. And here we are into our next fast episode. So, right, we do have some series going on as the YouTube live sessions. One is the fast series, that is your first aid simplified teaching, right? The USMLE First Aid book. Remember, when I say USMLE book, the content, the syllabus is the same, like you have for NEET PG, FMGE, or IIIMS. So, it remains valid for your these exams as well. And plus we have the other series, which is the fast 5 MCQs wala. Both of these you can find as playlist in YouTube, right? We have had some very important topics in the past that we have discussed in the fast series, the USMLE first aid teaching, like glomerular dynamics, high altitude changes, neurotransmitter changes, lymphatic drainage tricks, right? A lot of them you can check them. Plus the MedSynapse app subscribers, they have access to extra topics of fast as well, like you have the renal physiology formula, the CVS physiology formula, respiratory physiology graphs which are there. Plus in neurology, the stroke localization, spinal cord lesions, those are available as exclusive content to our premium subscribers of the MedSynapse app. You can see that fast folder which is there in the MedSynapse app, short course. So the subscribers can see those videos as well. In the today's video, we are going to discuss this concept of Lac operon. I keep receiving a lot of requests for this topic. As much as it seems difficult, before you understand it, actually it is not that difficult. So the purpose of this session is to cover this topic of Lac operon in a very easy way and a concise way, helping you understand and also remember, whatever is required for this topic of Lac operon. So let's start this. The PDF will be attached, like I said, in the short course fast folder. You will find, you can rewatch the video from there, right? Plus the PDF blank and annotated will be attached both to that video in that course, in that folder, fast folder. Okay. So first of all, tell me that, guys, how many of you find this topic difficult? Right, it is that one topic which is chota sa topic, but then if your concepts are not clear, you do not know the tricks, and if you get question in the exam, right, you cannot afford to miss a question on this topic, right? So how many of you find this difficult? Because at the end of the session also I'm going to ask you this question: do you still find this topic difficult, or now it is easy? So how many of you have tried reading this topic but could not understand it and you find it difficult? The moment you see a question on this topic of Lac operon, your first instinct is to skip the question. How many of you are like that? Yes? Okay, so let's see at the end that, how many of you, your views are changed after the session. So first of all, understand that Lac operon. Lac is the lactose, and operon, like the opera house, there is some entire orchestra which is playing, the orchestra of the genes which are arranged, which control some metabolism in a prokaryote. So remember, that is what is operon. Okay, so Lac basically here stands for lactose, and operon is this orchestra, the arrangement of the genes in the sequential manner which regulates this metabolism. Okay, so remember, this Lac operon is a classic example of genetic response to environmental change, right? So remember, this is genetic response to environmental change. The environmental change is how the levels of lactose are changing, how the levels of glucose are changing, and the genetic change is how is the transcription of these genes responsible for metabolism is changing. So genetic response to environmental change. Remember that in prokaryotes, E. coli, what is the preferred metabolite? It is glucose. If you give option of both glucose and lactose, obviously they will prefer using glucose, right? So, but when glucose is absent and lactose is available, so now when there is no glucose, only lactose is available. So, do the E. coli have any option? No. So, they will start using lactose, and to use this lactose, this lac operon will be switched on. Right, so lac operon basically helps in metabolizing the lactose. So when will it be activated? When the lactose is present and glucose is not present. Right, if both glucose and lactose are present, they will use glucose. Glucose is absent, lactose is present, then the bacteria will use lactose. Okay, so remember that in what state this lac operon is activated is when it's a low glucose state and it's a high lactose state, right? The glucose should be low and the lactose should be high. That is when it will be activated. Now, how does this low glucose and high lactose activate this lac operon? See, remember that when the glucose is low, how are the cyclic AMP levels? The cyclic AMP levels are increased. Now, understand few points about lac operon here. So, look at the genes which are there. These three genes which you see here, lac A, lac Y, lac Z. Okay, ये fashion में alphabetically arrange है, या ये fashion में देखो तो Z, Y, A, right? So these are called as structural genes. Okay, these are called as structural genes. So they encode for some enzymes required for metabolism of lactose. Then you have the lac I, right? The promoter and the operator, which are regulatory wale genes. Okay, they are the regulatory genes. So these are going to be the non-structural or regulatory genes, and these are going to be the structural genes. Now remember that you have the concept of this repressor, right? What is repressor? Repressor basically is a suppressor. Okay, in easy terms, remember wherever you read repressor, it means suppressor, right? It will suppress the lac operon. Now the question which comes in the exam is which gene encodes for the repressor protein? It is lac I, lac Z, lac Y, lac A. So please remember this question. It is lac I. How do you remember this? Learn this trick. It is I for inhibitor, right? I for inhibitor, repressor. Okay, that is how you will remember this. Is this clear with everyone? Right, so remember that I is inhibitor, that means it is repressor. So it is lac I which codes for repressor. Then you have the next question. From this DNA genes which are there, you know that what comes from DNA is RNA. What is that process called as? DNA से RNA जो बनता है वो होता है transcription, right? So transcription, for synthesis of RNA, you require this RNA polymerase. So at what site does this RNA polymerase bind? At which of these sites, you know, this is this lac I, promoter, operator, these genes, where does the RNA polymerase bind? So this one which you are seeing here, which is binding to the promoter, is the RNA polymerase. So look at this one, this orange color here is the RNA polymerase like you are seeing here. Remember P for P, it binds to the promoter site, okay? The RNA polymerase binds to the promoter site. Is this clear with everyone? Now you also need to understand that what promotes the binding of this polymerase, RNA polymerase to the promoter site is this cap, the binding of the cap to the cap site. Okay, so you have this cap here, the cap site where the cap binds, and it induces transcription by promoting the promoter site, the binding of RNA polymerase to the promoter. Okay, so let me clarify this. What is happening here is cap, okay, it binds to the cap site and it promotes the binding of polymerase to promoter. And then what happens? Then this transcription is induced. So this is the activator, the positive regulator, the positive activator of the lac operon is cap. Okay, so remember that cap, CAP, P for positive promoter, that is what you should remember here. Okay, lac I, repressor, is the inhibitor, it is the suppressor. Okay, and what activates this cap? High levels of cyclic AMP. When are these high levels of cyclic AMP present? When the glucose is low. Okay, when the glucose is low. So I hope now you can connect the dots. Remember it with a trick again. What you can remember is when should you wear a cap? Okay, when you should wear a cap. So the conditions when you should wear a cap is when the glucose is low, right? If you have hypoglycemia, wear a cap. And when do you get hypoglycemia? When you are on a camp. Okay. So remember, these go together. Low glucose increases camp, increases the binding to the cap, and it increases the promoter activity, the RNA polymerase binding to the promoter. So this is a positive activator. So imagine and visualize when you are on a camp, the low glucose levels are there. Wear a cap, right? Because that is what is promoted. Okay, it is promoted. So activating the promoter, right? Clear with everyone? So how does low glucose activate this Lac operon? By increasing camp, binding to cap, and then it is increasing the promoter activity. How does the high lactose activate the Lac operon now? Let's understand that. Now please try to understand this, that in Lac operon, if you see in the resting condition, how is the Lac operon? Is it repressed? Is it suppressed normally? Yes, right. So where does this repressor protein bind? This is the repressor. This is the repressor. So understand this, that the repressor is binding to the operator site. The repressor binds to the operator site. Okay? Now what happens when the lactose comes in? High levels of lactose converted to allolactose. It changes the shape of this repressor, inactivates it, and does not allow this repressor to bind to Lac operon. So this suppression is gone, right? This suppression is gone. So lactose is like inducing, it is switching on the Lac operon by inhibiting the inhibitor. Okay, by inhibiting the inhibitor. Is this clear with everyone? Right? So low glucose and high lactose. I hope this is clear now, that how does low glucose activate Lac operon? By increasing cyclic AMP binding to cap. Remember, they are similar in terms of spellings. So camp and cap go together. So cap acts at the cap site, increasing the promoter activity. And how does the lactose activate? Lactose inhibits the repressor. Okay. So that means it is inducing the Lac operon. Okay, it is inducing the Lac operon. Is this clear with everyone, guys? Let me know if there is any confusion so far. Is there any confusion here? Yes? So when this is the state, that is when the Lac genes are strongly expressed. Okay, that is when they are strongly expressed. Where do you have this cap site? Ahead of the promoter. Where is this promoter wala site present? Ahead of the structural genes. So if you look at the arrangement, it is Lac I. The cap is the cap for the promoter where the RNA polymerase binds. Then comes the operator where the repressor binds. And then at the end come the structural genes. So ahead of the structural genes, you all have this regulatory genes, okay, including the cap site, which is the cap for the promoter where the polymerase binds. Okay, that's great. I'm glad. Okay, so repressor protein, please remember, it binds the operator and it blocks the transcription. It is inhibited by lactose. So look at this. Lac genes are not expressed when the lactose is not there. Okay, so whether the glucose is high or the glucose is low, if the lactose is not there, the Lac gene will not be expressed. If the glucose is there, right, if the glucose is there, again it will not be expressed. Why? Because we don't want to use lactose, we want to use glucose. So all the time it is not expressed, or there is very low basal expression, right? But remember, it is strongly expressed when these two conditions are fulfilled, low glucose and lactose available. Both the cap and the lactose, the repressor wala thing, are acting together. So we have seen that Lac operon has how many structural genes? 3. There was Lac Z, Lac Y, Lac A. Lac Z encodes for beta galactosidase. You can remember this as galactosidase. Instead of sidase, you write it as galactosidase. Okay, so Z is for galactosidase. Y is the one which is responsible for entry. It is permease, the carrier protein which is allowing the lactose to go into the cell. Y is for entry, permease. A is for, remember, it is like TGTA, thiogalactoside transacetylase. So A for A, TGTA, Thiogalactosidase transacetylase. If at all you are asked question like this, you should know this. Regulatory genes is promoter. Remember P for P. The RNA polymerase binds here, right? The cap is present as a cap for the promoter. Lac operator, this is where what binds? The repressor binds. Okay, and the lac I, which encodes for the repressor protein. I is the inhibitor wala gene, which is encoding for repressor. Is this clear with everyone? Right, so this gives you the summary, and this is what we have already discussed. A quick review of this, that you have the structural genes, the regulatory genes, lac I encoding for repressor, binding to operator, right? Cap is the cap for promoter, where alanine polymerase binds. Cap is activated by this increased cyclic AMP. When do you get increased cyclic AMP? When the glucose is low. Lactose, when available, it changes the repressor so that it does not bind to the operator side. The repressor binds to operator side. Lac Z encodes for galactosidase. Lac Y is for entry, permease. Lac A is TGTA, thiogalactoside transacetylase. Okay, clear with everyone? Right? So understand this if the question comes. Lac operon is an example of negative control operon or a positive control operon. What is the predominant thing which is there, which is controlling lac operon? Is the repressor. Okay, the repressor, active repressor, which inhibits the transcription. So this is negative control operon. Another example of that is tryptophan operon. Like you have lac for leptose, you have tryptophan operon. Understand at least one difference in between the two. Now lac operon, when the lac, the metabolite lactose is present, what does it do? It switches on the lac operon. Opposite is for tryptophan. When the tryptophan is present, it represses, it suppresses the operon, it switches off the operon. So that means this one which you see here, right, this one which you see here is a repressible operon. Tryptophan is repressing the operon, and this one is the inducible operon, right? So normally what happens, this lac operon is switched off. When the lactose comes, then it is induced. Normally here this is switched on. When the tryptophan comes, then it is switched off. Okay, so at least remember this difference between lac operon and tryptophan operon. Both of them are negative control operons. That means they are regulated by the repressor. The active repressor is what is inhibiting the transcription. Right, in lactose, the presence of lactose induces the operon, inducible operon. In tryptophan operon, the presence of tryptophan switches off the operon, right? So that is repressible operon. So normally this is working. Tryptophan switches it off. Normally this is not working. Lactose comes, glucose is not there. That is when this is induced. Okay, that is when it is induced. Is this clear with everyone? The concept of lac operon, negative control, and it's an inducible operon, induced by the metabolite. And tryptophan wala is repressible operon, repressed by the metabolite. Okay, so that was the quick, concise explanation of the lac operon. Any queries here, guys? Any queries? Right, so if you want to quickly revise again with the one-liners, let's quickly revise this lac operon. First of all, tell me it is negative control or positive control? It is negative control operon, right? Is it an inducible operon or a repressible operon? It is inducible operon, induced by lactose. What are the structural genes? How many structural genes are there? There are 3 structural genes, which are lac Z, Y and A. Galactosidase, entry permease, and TGTA, thiogalactoside transacetylase. Which are the regulatory genes, the non-structural ones that you have? Lac I, promoter and operator. Lac I, promoter and operator. Lac I, inhibitor, is what is forming this repressor. What is the repressor binding to? Repressor binds to operator. Where does RNA polymerase bind? 5th point. RNA polymerase binds to promoter site. This promoter site is activated by cap. right? Cap is positive activator. What in turn causes this cap binding to the cap site? Increased cyclic AMP. When does the cyclic AMP increase? When the glucose is low. Okay, when the glucose is low. How does the lactose induce the lactose when present? How does it induce the lac operon? By inhibiting the repressor. Okay, by inhibiting the repressor, not allowing the repressor to bind to the operator. That is what all you need to know about this lac operon. So we have summarized this in the last slide. I hope this is clear with everyone. Any queries here? Right, so a topic that you would have read, maybe it would have taken 1 hour, 2 hour to understand and decode it. I hope we have done it in this 15 to 20 minutes. Right, for every topic that you learn, your homework is always to try solving questions on this topic. Right? Done? Any queries here? Similarly, like I said, we have decoded a lot of topics from the First Aid USMLE textbook, including physiology, pharmacology, pathomedicine के topics, ortho में bone tumors, patho में brain tumors, medicine में stroke localization, spinal cord lesions, right? Please do watch those sessions as well. They are available in Radsynapse app by Dr. Nikita. Like I said, the PDF for this will also be available in the Radsynapse app, the fast folder in the short course. Okay, right? So I hope none of you have the fear of this topic of lac operon now. And it is covered and it is easy, right? Tomorrow I'll be seeing the Radsynapse app subscribers for a live in-app live class for dermatology test discussion. The dermatology test is already live, which is the dermatology test is already live, will be discussed tomorrow at 12:30 noon, right? For the Radsynapse app subscribers, I'll see you there. Okay, thank you so much everyone, and I'll see you again soon in another YouTube live session decoding another difficult topic. If you find any topic difficult or you want me to simplify for you, please let me know in the comments, so that I can work on that topic in the next episodes of Fast, or just like that as well, we can simplify the topic for you with easy tricks and concepts. Okay, thank you so much. Have a great day. Keep studying, keep revising, keep med synapsing, and keep winning. Thank you so much.